Connaissance, recherche, sciences,
et sciences mathématiques,
théorie,
physique,
philosophie,
conférence,
archéologie,
nature.
Thank you, Philippe. Thank you, all the organizers, for this kind invitation and giving me the opportunity to present some of the work that I've been involved in the lab of Jean-Pierre Gorvel. So in the lab, we are interested in studying Brucella interaction with host cells, and I have been particularly interested in looking at interaction with dendritic cells and the role of these cells in the control of the host immune response during infection with Brucella. So I'll start with a very brief introduction on Brucella, since probably most of you don't encounter this pathogen very often. Brucellosis is still the most prevalent zoonotic disease worldwide, and with this slide I just want to illustrate that there's a number of Brucella species that infect a very wide range of both terrestrial and marine mammals. And in yellow I have highlighted the three strains that are virulent for humans: Brucella abortus, Brucella melitensis, and Brucella suis. And all of the work that I'm going to present to you today has been done with Brucella abortus strain. Now, this is still a problem in many parts of the world. There's many regions that brucellosis is still endemic, and obviously has important economic consequences for these countries. There are some vaccines available for animals, but there are many problems associated with these vaccines. Some are not very protective, and some are actually virulent in some animals. So there's still a need for development of better vaccines for animals. And there are growing new niches appearing. For example, the marine mammals, there's been reports of dolphin strains that are virulent now for humans being transmitted to humans, so this could become a problem. And also in countries where there is no longer brucellosis, like the United States and France, there are pockets of disease now appearing in wildlife. So just to give you an idea of the problem, in the United States, for example, at the Yellowstone National Park, more than 50% of the bisons are infected with Brucella. So this could become a problem in the future. Now, humans are secondary hosts. There is no vaccine available for humans, and it has a life of its own, my presentation. But there is an interest in developing a vaccine for humans, particularly in the context of using Brucella as an agent for bioterrorism, because Brucella is highly infectious via aerosol. For example, Brucella melitensis, 10 bacteria are enough to cause disease. So it could be important to develop a vaccine in humans. And although we can treat Brucella with antibiotics, it's quite a prolonged and difficult treatment. If it's not treated well earlier on at onset of disease, it can progress into a debilitating chronic infection, so it can have quite an impact. So humans get infected by ingestion of contaminated food products or contact with infected animals. For example, veterinary workers are at risk, or inhalation of infected aerosols. And central to all of the hosts is that Brucella is taken up by phagocytic cells and is able to survive and replicate within phagocytic cells. And then after uptake at the mucosal site of entry, then bacteria disseminate to the lymph nodes and spleen. And actually in humans, they can infect any organ. You can have severe endocarditis, you can have infection of the brain or the bones. So you can have quite severe complications if the disease is not, if the brucellosis is not treated correctly, and establishment of a chronic infection. This is not the case in the natural host. In the natural host, the disease is never fatal. And in fact, many of the animals can become chronic carriers, and they can carry bacteria and shed bacteria for their entire lives. And in fact, there is an interesting tropism for reproductive organs and also in pregnant animals for trophoblasts. So trophoblasts are also an important cell target for Brucella. And there's massive accumulation of bacteria in the placenta that eventually leads to abortion. Now, I've included in the slide the mouse model. Mice are naturally resistant to Brucella. but we can use them in the lab, some susceptible strains, to generate a persistent infection that can mimic some aspects of the disease. So we can recover bacteria up to 3 months post-infection, and we can see chronic inflammatory reactions in the spleen and liver. So it can be quite useful. We use it a lot, but we have to keep in mind that it's not really a natural host for Brucella. So you can look at the pathogenesis of Brucella. There's really 3 key aspects that are important. Brucella is inherently resistant to the host cell bactericidal molecules and activities. As I mentioned before, its ability to replicate and survive within host cells is really essential for its virulence, and also its ability to hide from the host immune response. And it seems that in many cases actually control and modulate this host immune response to cause disease. And in our lab, we've been doing a lot of work especially in these 2 second aspects of the infection. But most of the work has been done using cultured cell lines, non-phagocytic cell lines, or macrophages. And so I've been interested in looking at dendritic cells. And this started with some experiments that I was doing in the lab with Hugues Leloir when he joined the lab, and we were infecting murine intestinal loops with Brucella, and we could see bacteria readily being taken up by CD11C positive cells that were in the subepithelial dome of the pire patches. They're also MHC class II positive, so dendritic cells that I'm not showing you here. And so I decided to set up an in vitro system using murine bone marrow derived dendritic cells. These are differentiated in vitro with GM-CSF to look more closely at this interaction with Brucella. And we can see Brucella efficiently replicating within these dendritic cells. Replication was dependent on the type 4 secretion system. This is a type 4 secretion system mutant, VRB, that has been shown to be essential for replication in all the other cell types analyzed so far. And it replicates within the endoplasmic reticulum, just as has been described for macrophages. And this is nicely illustrated by electron microscopy. We can see this is the bacteria within a vacuole. We can see ribosomes outlining the vacuolar membrane. We can detect glucose 6-phosphatase, which is an ER lumen enzyme, and this accumulates nicely in the vacuolar membrane of replicating bacteria, and also immunogold labeling with an anti-calnexin antibody. So this seemed like a good model to look at Brucella infection of dendritic cells in vitro, very similar to macrophages. So the first question, an important question we wanted to ask, was, does Brucella infection have any effect on the maturation of dendritic cells? Now, this is a complex process, maturation of dendritic cells, that involves many changes. Here I'm just illustrating MHC class II labeling in an immature dendritic cell. MHC class II is distributed intracellularly, and when dendritic cells, for example, encounter a microbial stimuli, they undergo this process of activation, the maturation. And you can see there's a striking change in morphology. There's upregulation of MHC class II, now moves to the cell surface. There is appearance of these dendrites. There's also increased surface expression of co-stimulatory molecules, secretion of cytokines, and eventually, if they're fully mature, there's also efficient antigen presentation. So we wanted to know what was the effect of Brucella on dendritic cells during infection. One of the things we looked at was the formation of DALIs. Now, DALIs are transient aggregates that form during the process of maturation, the aggregates of poly and monoubiquitinated proteins that appear soon after the induction of maturation. You can see them here labeled in red, these large structures labeled with an FK2 antibody. You can see them very well in salmonella-infected dendritic cells. And in the case of Brucella-infected dendritic cells, there's either no DALIs, or if there are some DALIs, they're much smaller. So suggesting that there is no induction of maturation of dendritic cells or low levels. But most importantly, if you looked at MHC class II distribution, you see that the Brucella-infected dendritic cell looks very similar to a non-infected cell, in which MHC class II is distributed intracellularly, in contrast to Salmonella, where you can have higher levels of expression of MHC class II at the cell surface, and even more strikingly on E. coli LPS, fully treated, fully mature dendritic cell. There's a lot of MHC class II on the cell surface, and there's also collapse of LAMP1 positive lysosomal compartments at the microtubule organizing center. So by microscopy, we could see that the Brucella-infected dendritic cells were not significantly maturing. We also look by flow cytometry, and we could see surface expression of co-stimulatory molecules such as CD40 and CD80. You can see much lower surface expression of these molecules compared to Salmonella-infected cells. It's not, there is some induction compared to mock infected. You can see it by the shapes of the curve in gray, in shaded gray. It's an intermediate phenotype, so there is some induction of activation, but not full activation, and also MHC class 2. We do not see any difference in MHC class 1 expression. And also cytokine secretion. We could see Brucella-infected dendritic cells secrete very low levels of pro-inflammatory cytokines like TNF or IL-12, interferon beta, and also IL-6, compared to, for example, a heat-killed Brucella. Now, one of the important questions is this important for Brucella replication? And so to do that, we decided to add, for example, E. coli LPS soon after infection, 30 minutes after infection, to induce maturation of dendritic cells. And in this case, we completely inhibit growth of the bacteria inside the dendritic cell. And it's only when you add E. coli LPS 12 hours or after that you start not to have an effect on the Brucella replication. So this says that Brucella needs the dendritic cell to be in an immature state in order to reach the endoplasmic reticulum and start to replicate. So it is important for its virulence. So we went on to identify a Brucella protein that we call BTP1, Brucella tear-containing protein. As Philippe mentioned, it's important in the control of host immune response in Brucella. And so this protein has a tear domain. Tear domains are present on all toll-like receptors and the adaptive molecules and are essential for toll-like receptor signaling. And so we made a mutant. We were a bit disappointed because the mutant replicated just as efficiently as the wild type. But we could see that in dendritic cells infected with the BTP1 mutant, there was higher levels of MHC class 2 surface expression and also higher levels of cytokine secretion. You can see here particularly TNF alpha. So suggesting that it was playing a role in control of maturation. But this was all very nice, but it was done in vitro, in cultured dendritic cells. And as we know, in vivo, there are many types of subsets of dendritic cells. There is not just a single one. So we've been trying now more recently to go in vivo and look at the role of dendritic cells and BTPs in infection. So this started when Christelle Archambault joined the lab of Bernard Malissel, just next door to us. And she was interested in looking at lung infections. And so in collaboration with Hugues Leloir for the microscopy of tissue sections, we infected mice intranasally with Brucella abortus and looked at the lung. And we could, in this case, we used Brucella abortus expressing the S-red, so it's the red dots that you see here correspond to the bacteria. And we can see Brucella replicating very nicely in lung cells, in this case positive CD11C. Now CD11C in the lung is expressed in both alveolar macrophages and dendritic cells. But we found that 99% of the infected cells were alveolar macrophages. They expressed lysozyme and they were MHC class 2 negative. So most of the bacteria had been taken up by alveolar macrophages. When you look at dissemination to the draining lymph nodes, then you see both macrophages, which are here lysozyme positive, but also MHC class 2 high lysozyme negative cells being infected, and these are dendritic cells. And a significant proportion of those correspond to CD103 positive dendritic cells that were seen in the draining. This is sections from mediastinal lymph nodes, in fact. And so dendritic cells were capturing bacteria and taking them to the lymph nodes. And to study better the role of dendritic cells, Christelle then went on to deplete alveolar macrophages, or partially deplete them, with clodronate-loaded liposomes. And in this case, then we could detect a significant proportion of the infected cells were dendritic cells in the lung. So we could increase the number of dendritic cells infected. They were MHC class 2 high and CD11C positive. And when this happened, then we saw a much higher replication of bacteria in the lung early on when you deplete macrophages, so this corresponds to these lighter colored circles. And you can see much higher levels of bacterial counts in the lung and also higher dissemination to the liver and spleen. So the macrophages are really important to contain the infection in the lung. And obviously what we wanted to look at was the role of BTP1 in dendritic cells. And so we went on to infect mice with the BTP1 mutant, and what we found was that in specifically subsets of dendritic cells, we could see higher levels of CD80 and CD86 being expressed in the dendritic cells obtained from mice infected with the BTP1 mutant compared to the wild type. And very interestingly, this was only observed in CD103 positive dendritic cells. There was no difference in CD11B. So this tells us that actually the different subsets of dendritic cells might be very important. So now we have to do a lot more work to characterize these subsets further. And the story is not as simple. We have recently identified, in looking at the genome sequence of Brucella, a second-tier containing protein that we call BTP2 that is also contributing to the control of the dendritic cell maturation. So a BTP2 mutant, just like a BTP1, has higher levels of MHC class II distribution. It also has higher levels of cytokine secretion and DALIS formation. And also, you can look at, if you look at NF-kappa B translocation in infected cells, so you can see in wild type, in the negative, you don't get NF-kappa B in the nucleus. In salmonella infected cells, very early on, at 2 hours, you can already see NF-kappa B translocated to the nucleus. And in the case of BTP1 and BTP2 mutants, you get higher percentage of cells with NF-kappa B in the nucleus compared to the wild type, and this is even higher in the double mutant, suggesting that these two proteins may be acting together to control signaling within dendritic cells. We haven't done the lung infection studies yet with these double mutant, but we have started infecting mice and looking at, we were looking at survival, but they are just as virulent as the wild type. But what we found was that at late stages of the infection, this corresponds, open bars correspond to 60 days, you could see a much higher number and size of granulomas being formed in both the liver and the spleen when mice were infected with the double mutant BTP1, BTP2. So really consistent with the idea that these proteins are really playing a role in the immune response during infection. And I just want to end by saying that we think these two proteins are acting in different pathways. And to do that, now we are moving to more in vitro systems. In this case, I'm going to show you transfecting cells, cultured cells, with a specific TLR and an NF-kappa B luciferase reporter system, and then add the appropriate ligand, for example, in this case PAM, and you can see, you can measure luciferase activity. And you can see that in increasing concentrations of BTP1, there is an inhibition of TLR2 signaling and also TLR4 signaling, whereas there's no effect on TLR9. In contrast, BTP2, it's very efficient at blocking all TLR pathways that we have tested, TLR2, TLR9, TLR4. And recently there's been a paper suggesting that BTP1 is actually increasing the ubiquitination and degradation of an adapter molecule, TYRAP, MAL, which is specific to TLR2 and TLR4 pathway. We haven't seen these, been able to reproduce that in dendritic cells, but we do see that BTP1 is competing with MAL and TYRAP, whereas BTP2 is not having the same effect. So they seem to be acting on different pathways. So in conclusion, not only macrophages, but also dendritic cells seem to provide an intracellular niche for Brucella survival within host cells. And we've identified two proteins that seem to contribute to downmodulation of dendritic cell maturation by interfering with specific toll-like receptor signaling pathways. And I just want to end. I think I've mentioned everyone that was involved in the work throughout the talk. I just want to add that this work was done in collaboration with 3 labs at the CIML, Philippe Pierre for the dendritic cells, Alena Alexopoulou for the TLR signaling, and Bernard Malison for the lung infection model, and also with a lab from Diego Comerci in Argentina for all the BTP work. And I thank you for your attention.